Good day, this is the Professopher, drwillphd, PhD, and today's Wednesday, January 25th, 2023. It is 5:44 and 43 seconds, a.m. Eastern Standard Time, and today's research, the genome. So let's hop to it here. The Research Scientific Genome Editing Procedures Optimized, date January 24th, 2023. Source: Heidelberg University. The scientists succeed in boosting the efficiency of CRISPR-Cas9 and related methods and modifying, initially, inaccessible DNA sequences. The research scientists fine-tune these tools to enable effective genetic screening for modeling specific gene mutations. The genome editing means the deliberate altering of DNA with molecular genetic methods. It is used to breed plants and animals, but also in basic medical and biological research. The most common procedures include the gene scissors, CRISPR-Cas9, and its variants, known as base editing. In both cases, enzymes have to be transported into the nucleus of the target cell. Upon arrival, the CRISPR-Cas9 system cuts the DNA at specific sites, which causes a double-strand break. New DNA segments can then be inserted at that site. Base editors use a similar molecular mechanism, but they do not cut the DNA double strand. Instead, an enzyme coupled with a Cas9 protein performs a targeted exchange of nucleotides, the basic building blocks of the genome. In 3 successive studies, the team succeeded in considerably enhancing the efficiency and applicability of these methods. A challenge when using CRISPR-Cas9 consists in the efficient delivery of the required Cas9 enzymes to the nucleus. The cell has an elaborate bouncer mechanism. It distinguishes between proteins that are allowed to translocate into the nucleus and those that are supposed to stay in the cytoplasm. Access is enabled here by a tag made up of a few amino acids that functions like an admission ticket. The scientists have now come up with a kind of generally valid, very important admission ticket, which lets enzymes equipped with it into the nucleus very quickly. They named it the High Efficiency Tag, HAY tag for short. Other proteins that have to penetrate the cell nucleus are also more successful with the HAY tag. In a further study, the scientists showed that base editors operate highly efficiently in the living organism. They are even suited to genetic screening. An experiment with Japanese rice fish, they were able to show that these locally limited, targeted modifications in individual building blocks of the DNA achieve an outcome that is otherwise only obtained by the comparatively laborious breeding of organisms with altered genes. The research team focused on certain genetic mutations. These mutations were suspected of triggering congenital heart defects in humans. And to modify individual building blocks of the DNA of relevant genes in a model organism, the scientists were able to imitate and study fish embryos with the described heart defects. The targeted intervention led to visible changes in the heart already during early stages of fish embryonic development. That enabled the researchers to confirm the original suspicion and establish a causal connection between genetic alteration and clinical symptoms. The precise intervention of the genome of the fish embryos was made possible through a specially developed software, AC of the bases, which is available online. It allows for identifying genetic locations that very efficiently lead to desired changes in the target genes and the resultant proteins. The scientists say that the Japanese rice fish is an excellent genetic model organism for modeling mutations like those identified from the respective patients. The research method enables an efficient screening analysis and can therefore offer a starting point for developing individualized medical treatment. A third study deals with the limitations of base editors. For such an editor to bind the DNA of a target cell, there has to be a certain sequence, motif. It is called protospacing, adjacent motif. And if this motif is lacking near the DNA building block to be changed, it is possible to exchange nucleotides. As the two base editors in a single cell are used in succession, the initial step, a new DNA binding motif for a further base editor, is generated, upon which the second editor, which is applied simultaneously, can edit a site that was inaccessible before. And this staggered use turned out to be highly efficient. With this trick, the scientists were able to increase the number of possible application sites of established base editors by 65%. Now DNA sequences that were initially inaccessible can also be modified, as optimizing the existing tools for genome editing and their fine-tuned application results in enormously varied possibilities for basic research and potentially novel therapeutic approaches. And this is the Professopher, Professor of Microbiology, Bioinformatics, and Pathophysiology. This video was created for educational purposes only. Hope you enjoyed listening. Until next time, have an awesome day.